Welcome back, everybody, to this final part of membrane potentials. In this video, we're going to be talking about action potentials. I've touched on this a couple of times in the previous videos, kind of that we would get to this point. This is really kind of what I would call the meat and potatoes of this specific PowerPoint. So, what is an action potential? So, an action potential are the actual messages that our body is going to be using to send along those nerves and to the muscles and up to the brain to communicate what is happening. Okay? This is actually how we transport that that signal throughout our entire body. So there's going to be a rapid rise and then a fall in voltage that runs across a cellular membrane. And we're going to kind of take a look at kind of how that happens and also what that specifically looks like. So this is what we're going to see when we look at an action potential. We're going to see this little graph. Because remember that our resting membrane potential is negative 70 millivolts. Now, of course, remember right here when we look at this line right here at the negative 70 resting membrane potential, that the sodium potassium ATPase, that's what's helping with our resting state. And we're going to look in at how this whole thing happens and then also a little bit why it's so important. A little bit of vocab before we we continue. We've already looked at a couple of these words. We have depolarization. This is going from a negative value becoming more positive, heading towards zero. And we also have repolarization, which is going from that positive from zero going back down to our normal negative value. We have hyperpolarization. Hyperpolarization is what happens when we go lower than our resting membrane potential. And we also have overshoot, which is when it goes above that zero millivolt value. So we get a little bit of overshoot on the top end and a little bit of hyperpolarization on the bottom end. Last one, probably one of the most important, is threshold. The threshold is a specific value for these different cells required for an action potential to happen. There's a really nice slide right here, kind of about the basics of the action potential. The most important thing that we need to know is that an action potential is an all or nothing event. Okay? As soon as we reach threshold, boom, an action potential happens. So as soon as we get to this negative 55 value, boom, we're going to have the rest of the process occur and come back down and then eventually reach back to resting membrane potential. If we do not reach threshold, we do not have an action potential be generated. Super, super important to understand that. Additionally, our action potentials are going to have a constant amplitude. So that height value is going to be constant. We're not going to get like small action potentials or bigger action potentials. No, it's always going to be the exact same. It either happens or it doesn't, and it's the same amplitude every single time. It's initiated by depolarization, which we said going from negative, we're going towards positive. That's depolarization. And it involves changes in permeability. When we talk about permeability, we're talking about what is allowed to pass through the membrane. And we'd mentioned those sodium and potassium gates. Those are our voltage-gated channels. We're going to come back again and look at these in just a little bit when we get to those steps. So here we can see this is that all or nothing principle. We can have little guys here. As soon as it hits threshold, boom, action potential. Okay? We have talking about the permeability, we have at different part. We'll talk about this as well. When we have different points of the action potential that occur, we are more or less permeable to specific ions depending on which channels are open. and it's relying on those voltage-gated ion channels, which we'll get back around to. Okay. Here's all this is used for that long-distance communication. So non-decremental, it's a good word to use. It doesn't fizzle out. Once it goes, it's all or nothing. It's the same height. So we're going to go kind of the step-by-step here of the action potential. And I'm going to add a fair bit of information into this little graph that we have. So we start with resting membrane potential. Remember, for the neuron, that's negative 70 millivolts. I keep saying that because it's important to remember and keep those differentiated. Just because if you look at a graph and you see it's at negative 70, immediately you can know, okay, we're talking about a nerve cell. If you see that it's at negative 90, okay, we're talking about a skeletal muscle cell. If it's at negative 60, okay, we're more likely talking about a smooth muscle cell. You can tell that just by looking at the graph. Step 2, we have a depolarizing stimulus. Now, this can be a whole number of things that cause this depolarizing stimulus. It can be from something like mechanoreceptors, like based on touch, feeling, that sort of stuff. It can also be something like leaky channels. You can have leaky channel stimulus, not always necessarily for nerve cells, but more for like cardiac tissue. But you need to have some sort of stimulus which is going to begin the depolarization, which gets us from that negative 70 value up to threshold. Okay? Understanding, so here at this point at 2, we have sodium channels get opened from some sort of stimulus. I'm going to add here at 2. And I say sodium, it's mostly sodium because that's one of the most abundant. So sodium channels are open, allowing for depolarization. And these are not necessarily gated sodium channels. Specifically, understand that these are not voltage-gated channels. Okay, that's going to come later. These are not our voltage-gated channels. They're just regular sodium channels. Are going to allow for some of the beginning depolarization, specifically to threshold. Okay. Good. Step 3. As soon as we reach threshold, okay, and you'll see kind of all over. You look at different charts and graphs of all of this, and different charts and graphs say different things about this. I will go with what this professor is saying right here, but understand that there's a little bit of disagreement, kind of exactly when these sort of like the channels, when exactly they start to open, start to close, all that kind of stuff. So you'll see different graphs kind of all over. Pay attention to what your specific teacher, your specific professor says about this, and follow whatever it is that they specifically say. So as soon as we reach threshold, we have voltage-gated sodium channels and the voltage-gated potassium channels begin to open. Now, jumping back, and we saw here, right? We can be a little bit more specific with this. Okay? So at point 3, and I want you guys to I really highly recommend printing this out and writing all of this down at each of these different spots so you have a quick kind of cheat sheet to look at as you're reviewing and as you're studying, and just also for practice. So at point 3, we have, we're going to go very, very specific. We have the activation gate of voltage-gated sodium channels opens. Then we also have voltage gated potassium channels start to open. Okay, both of these are occurring right here. Things to remember: the potassium channels, these are really slow, so they take a while to fully open. So what does this mean is happening at this point? Right now, big idea that we're going to focus on is that the cell's permeability to sodium goes up, or it increases, because we're going to be letting a lot more sodium come into the cell. So the permeability to the sodium goes up, it increases because we're allowing all this sodium to come into the cell. And that's how we get this spike going up. We get depolarization. Depolarization begins here at 2 and goes all the way up to this peak. This whole chunk is known as depolarization. Okay? Next step, 4, we get that rapid sodium entry, depolarizing the cell. Kind of 3 and 4, as soon as that opens at threshold, all the sodium rushes in, and we have that increased permeability to sodium. We go up, 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 just like that. Remember, from 0 above, that's called overshoot. Depolarization, the whole thing we call depolarization. The 0 to 30 part, more specifically, we call overshoot, though. Next step, number 5. Our sodium channels close, and the slower potassium channels are finally open. So again, a little bit more specifically, the inactivation gates of voltage-gated sodium channels closes at this point, and the voltage-gated potassium channels are fully open. I like to write all of this out because it helps keep everything straight with knowing that it's a voltage-gated sodium channel. You can abbreviate this a little bit if you want. You can do something like voltage-gated sodium channel, something like that, if you want to keep it a little bit simpler. But I highly recommend writing the whole thing out, at least for the first couple times that you're learning this, just so that you get in the habit of writing the full channel name, because when we start to look at other graphs like this in skeletal muscle and cardiac muscle and smooth muscle, we can start to get a lot of mix-ups with the names and what specific channels are where. So I like to write out the full names just to be safe. 5 right there. And so when the potassium channels are now open, right, what's going to be happening? We know that the potassium wants to come out of the cell because of its chemical gradient. There's a whole lot of potassium on the inside, not a lot of potassium on the outside. So we get, let me find the one slide. There's a nice slide here. Here we go. So during the downstroke, our potassium permeability increases. So potassium permeability increases because our potassium is able to move through the membrane, even though it's moving out of the cell. Remember that the permeability just means that if it can cross the membrane. Okay, so because it can cross out super easily, that's what we're focusing on. And then our sodium permeability decreases right there. Okay, these are really, really good test questions to ask. Like, okay, he'll give you the graph, and he'll point to a spot on the graph, and he'll say, at this point, what is occurring with the permeability? And knowing, okay, where is sodium permeability increased? Sodium permeability is increased all along here. Whereas sodium permeability decreased all along here, whereas potassium permeability increased all along there. Okay? Easy test questions that they can ask. Okay? Now we're going to see, as soon as those are open, we have the potassium moving from the cell to the extracellular fluid. This is our repolarization that is occurring. Okay? So resting membrane potential, and that is our initial depolarization due to just regular sodium channels. Then we get full depolarization because of those voltage-gated sodium channels. Sodium channels close, voltage-gated sodium channels close up here. The potassium voltage-gated channels fully open, and we come back down with repolarization. This part over here is the overshoot, and then we continue all the way down, and we get hyperpolarization. So the potassium channels remain open, additional potassium leaves cells, hyperpolarizing it. And again, that's because that potassium gate that we saw over here, this gate, it moves pretty slow. So it slowly closes and allows potassium to continue to leak out a little bit longer than it should. That's that hyperpolarization. The reason why this occurs, the reason why we get this hyperpolarization, is because we don't want the body sending too many signals too quickly. Okay, we kind of want to regulate that control, and that's what that does. We get what's something that is called an absolute refractory period, and we get a relative refractory period. What these are, this is a portion of the action potential where another action potential is impossible to happen. The relative refractory period is that an action potential may happen, but will require a much larger stimulus to reach threshold. Okay, and I know that I have a couple of slides. If you have my notes, I've included, which I'm going to be updating the ones that I have here, of these ones that I'm adding things in with these videos, as well as my previous notes as well. There are several slides on that clearly show this. Let me see if I can find one of the slides that specifically show this period. Here we go. Right here. It shows in a different type. It's a little bit of a different type of graph, but we have an absolute, and then we have an absolute refractory period and a relative refractory period. Oh, so yeah, this whole refractory period, the whole thing, absolute versus relative. So same here. I would mark it from as soon as we hit threshold to right here. This is our absolute refractory period. And then after that, until we get back to our resting membrane potential, that is considered our relative refractory period. And again, that is specifically in place to make sure that we don't have overstimulation of the cells. We don't have action potentials kind of layering on top of each other, sending too big of a message. Okay, so here we go. The voltage-gated potassiums fully close, and less and less potassium leaks out of the cell. And as well as we continue to go along this way, we have something called that sodium potassium ATPase, that pump, sodium potassium pump, that's working. It's pushing 3 sodium out, bringing 2 potassium back in to help level us back out to our resting membrane potential during this time. So finally we get back to our resting membrane potential, and we can have another action potential occur as long as we get another stimulus. The rising, the falling, the after hyperpolarization, and back to resting. Here we've already talked about this, about our sodium gated versus our potassium gated, kind of how all of these happen. These slides do a pretty good job of laying it out specifically, step by step. So I recommend looking through these slides as well. This is one, if you remember back in Biochem 1, we talked about negative feedback loops, where something would loop back once we created a product. Something would loop back and stop the process from happening. The action potential are going to have the opposite. They're going to have what's called positive feedback. Okay, the positive feedback, also known as a feed forward loop, we can see it here where when sodium enters the cell, we get more depolarization, which then more of the sodium channels activation gates are going to be opening, which leads to more sodium entering, and so on and so on and so on. And so the actual depolarization of the neuron in this case is a positive feedback loop. Here's the refractory period, again, that we already talked about. One of the things I didn't mention is that it prevents the retrograde movement of the action potential, so it prevents us, from all this happening. So, like, for example, if we're in, because as we know, neurons are going to connect to each other. So we have a soma, which is a neural cell body. We have an axon, and then we have kind of all this, the axon terminals coming out, as well as the body has these dendrites. These can go and they can connect to other somas and spread the message like so. So if we have a message, let's say the sensors get picked up right here, and we want to send it this way towards the brain, we don't want retrograde movement of the action potential. We don't want it going the wrong way. So that refractory period also assists with that to make sure that the message only goes the appropriate way that we want it to go. Again, it's absolute versus relative. Here's the picture. He hasn't moved over a little bit after the hyperpolarization, but yeah. Okay. This video is getting a little bit long, but we'll go ahead and finish it out. Action potential conduction. So as soon as we get the action potential occurs, it needs to, as soon as the action potential occurs in one spot, everything that we've talked about, all these voltage gated, all these things, just like we saw when I drove the soma, the cell body, and the axon, we're talking about just this one specific point where this is occurring, where this is all happening. Now we need that signal to travel. And so what's literally going to happen is the portion of the cell, or the axon in this case, right next door to where this happens. So like let's say we started right here. So we get that stimulus that starts right there. We get it gets up to threshold and the sodium leaks in, everything happens. So if we have sodium leaking in, kind of what's that's going to do, that sodium is going to leak and dissipate sideways. And when it goes sideways, it's going to then activate the channels that are next to it. And then this right here, that's going to have an action potential occur, where we have that sodium channels opening and the potassium channels and so on. And then the sodium that's let into the cell from here moves over and does to the next part. Then it moves over and goes to the next part. It moves over and so on. And that's actually how the message travels. We can see this kind of a little bit more broken down here in these next couple of slides. This is a lot of kind of mumbo jumbo kind of stuff. It's pretty it is pretty clear if you go take the time and read through it. I'd recommend just kind of read through each of these portions. I already explained the main ideas specifically, but yeah. And then these are some of the things that can affect the action potential: conduction velocity, velocity being how fast this signal travels. These are going to be a couple of test questions, especially this one: diameter of the axon. With a larger axon, we have a faster signal that goes through. The longer the nerve, obviously, the longer the nerve it is, it's going to take a little bit longer to get to wherever it needs to go. So, like, for example, and when we talk about longer and time-wise, like, these are still happening incredibly fast. Like, this is still a matter of milliseconds that kind of all of this is occurring. And then additionally, we throw in the myelin, remember, along our axon. Let's start specifically in the peripheral nervous system, so outside of the brain and spinal cord. We have Schwann cells, which myelinate. The myelination, what it actually allows for is what's called saltatory conduction. Saltatory means jumping conduction. So we have our action potential occur right here. We get the whole sodium channels, potassium channels, depolarization, repolarization, all that happening. And then the sodium that's here can float all the way over here and cause, bam, action potential there. Pass it, it hits another myelin sheath, and it can jump and skip. So that way it doesn't have to go through the whole entire process all the way along the axon. It can jump and jump and jump, and that makes this process much, much faster. Remember, these guys in the peripheral nervous system, known as the PNS, these are our Schwann cells. In the central nervous system, so the brain and the spinal cord, they are oligodendrocytes. In future classes, you'll start to look at medical conditions, where we get degradation, we get destruction of these types of cells, as well as some of our other nerve disorders, things like crush syndromes. You'll learn about something called Saturday night palsy. It gets that name from somebody who's been out drinking a lot Saturday night, and they fall asleep kind of with their arm over a bench or a chair, the back of a chair, and it literally presses and compresses some of the nerves in the axilla. And with that crushing force can damage some of the Schwann cells. And if those get damaged, you lose this nice conduction. And that's what causes pain and nerve type symptoms within the body. And that eventually does, that can, as long as the damage is not too extensive, it can heal, and the Schwann cells can kind of restore that area. That's a little extra tidbit. You'll talk a lot about those conditions and stuff in future courses. Some alterations: local anesthetics, lidocaine and procaine, for example, they bind to the sodium channels. If they're bound to the sodium channels, then the sodium channels can't open. If those sodium channels aren't able to open, then we're not going to get pain signal or any signals going to the brain. Same thing with those local anesthetics. Yeah, there's just another little chart showing it doesn't let us reach threshold. It actually drops us lower. It drops us lower. So we need a larger stimulus in order to have any kind of action potential occur. Okay, I know this video was a little bit longer. Big main ideas I want you to really focus on is really understanding this whole process, exactly what happens in each step going forward, what happens where, the definitions of everything, because this is going to set us up. If you understand this super well, and write yourself out a nice graph, a nice chart like this. I would do it by hand because you'll remember it and retain it a lot better. If you do that, you're going to be much, much better off and in a much more comfortable place to learn the next steps going forward when we start looking at other cells. Okay, thank you so much for watching, guys. Have a great day, and as always, if you have any questions, comments, concerns, anything going on, please drop them down below, and I'll get to them as quick as I can. Have a good night. Bye.